Thank you very much, dear Chair, dear colleagues. Can you hear me?
Yes.
Perfect. Thank you very much for this kind invitation to participate in this wonderful conference. It's a great honor to share with you the latest developments in STAR, Stereotactic Arrhythmia Radioablation Therapy, which is a novel, non-invasive approach to treat ventricular tachycardia in selected patients. My name is Tomasz Jadczyk. I am a cardiologist by training and electrophysiologist by practice. Today, I will explore why STAR therapy works, and I will also demonstrate how we can leverage tools such as OpenEP and EP Workbench to measure STAR effects on human myocardium. So let's go for it. Ventricular tachycardia in patients with structural heart disease remains a major therapeutic challenge. Recurrent VT is associated with increased risk of sudden cardiac death, progressive heart failure, frequent hospitalizations, and also impaired quality of life of our patients. So what patient says with ICD and shocks, it's like a horse just kicks you in your chest. The other would say it's like a dynamite exploding inside. In numerous cases, frequent ICD shocks continue even with optimal medical treatment, underscoring the ongoing need for effective therapeutic strategies. Guideline-based management combines antiarrhythmic drugs and catheter ablation, which is, of course, an established treatment, reducing arrhythmia recurrences and also ICD shocks. However, conventional RF ablation is limited, and this strategy is limited in efficacy with a recurrence rate up to 30%. Obviously, there is a need for a better care in patients with VT. I believe that the most remarkable breakthroughs happen when we speak across disciplines and translating radiotherapeutic experience. A novel treatment strategy has emerged for patients with intractable VT, so arrhythmia which is refractory to antiarrhythmic medications and also conventional RF ablation. So this is how the STAR is born for selected patients with VT. Here you can see the TrueBeam system, which is a linear accelerator. In STAR procedures, this technology enables non-invasive ablation of arrhythmogenic tissue by delivering a single high-dose radiation, and usually it's 25 gray in one fraction, to a precisely defined target. The radiation beam, as you could see on this animation, can be modulated in shape and intensity, allowing for high conformal dose delivery, which is like perfect for complex anatomy of the heart. During treatment, the C-arm gantry rotates around the patients, delivering radiation from multiple angles, as you can see here. So the full dose, this 25 grays, is typically administered over 2 to 3 arcs, ensuring that the target receives the therapeutic dose while we are saving the organs at risk all around the heart. So this is the general strategy. STAR has been used for patients unresponsive to conventional therapy since 2012. As I mentioned, it reduces the burden of ventricular arrhythmias and ICD shocks. If so effective, at least in short observations like 6 months, we wonder how it works. In animal models, STAR is ablative by triggering fibrotic response, like transmural fibrosis, over weeks and months. The next question is, of course, how is it in humans? You can see here results of the early studies in human. Like, no doubt, STAR offers very promising non-invasive treatment that reduces ventricular arrhythmias by more than 85%. Here, you can see histopathological sample of human post-MI heart, which was treated with STAR. So what we can see here, of course, is a dense scar from previous myocardial infarction. scar border zone, and viable myocardium. The question is, where is transmural fibrosis that was expected from the animal studies? So, of course, the first question is, why does it work, and why does it work so fast? We still don't fully understand how STAR produces such a rapid antiarrhythmic effects. And it becomes like clear and more clear that it's not just about radiation-induced fibrosis. So to kind of set the stage, I would like to walk you through a few key concepts that give us some insight into our understanding of STAR mechanisms. For the STAR, we use ionizing radiation. So first, let's meet the dark side of ionizing radiation. Radiation affects heart cells in two main ways. First, directly by damaging DNA, and also indirectly through oxidative stress. This oxidative damage can change gene expression. It leads to protein alteration, cell death, genomic instability. What's particularly important for us is that the production of reactive oxygen species, ROS, can increase heart susceptibility to focal activity and reentry. So, two key mechanisms responsible for triggering arrhythmia. Just take a look on the broader concept, how radiotherapy may impact heart. Please see how much actually negative impact we can find. Almost all components of the heart are impacted by ionizing radiation, and apart from arrhythmias, it impacts valvular structures, vasculature, also even pericardium. However, the consequence of ROS production after targeted irradiation is perhaps to trigger a compensatory downstream molecular response, leading to the production of mitochondrial antioxidants, ultimately giving an antiarrhythmic effect. So it's like a very dark and very wide power in one kind of treatment strategy. And in addition to structural effects, radiation also impacts the electrical properties of heart cells. It modulates ion channels like sodium, potassium, calcium channels, as well as it works through gap junctions like connexin 33 and also calcium handling mechanisms. These early changes may explain the antiarrhythmic effects of STAR, as they represent form of electrophysiological reprogramming that occurs before fibrosis develops. This is a pretty busy slide, but let's focus on the key elements here. STAR impacts both cardiomyocytes and stromal cells. With acute and early response, we talk here about minutes, hours, and days, and also the late response in months. Translating this busy slide into something more easy to capture, I would like to divide STAR mechanisms in two groups, early and late effects. For early effects, we can see electrophysiological reprogramming. This is mainly through connexin and sodium channel. We can see improvement of ejection fraction in some patients. However, the late effect is mostly focused on fibrosis, even conduction blocks. And we can see mitochondrial dysfunction, endoplasmatic reticulum stress, and also involvement of inflammatory cytokines. Please notice that inflammation and oxidative stress take role in both early and late effects mechanisms. So apparently this is a molecular path worth exploring, especially for the electrophysiological reprogramming. Let's take a closer look on the electric reprogramming through this interplay between sodium, potassium, calcium channels, also connexin 33. There is a suppression of abnormal automaticity, then suppression of triggered activity, altered conduction velocity, and also prolongation of ERP. Cumulatively, these mechanisms suppress re-entry loops and stop ventricular arrhythmias. Conduction velocity concept is of special interest to our research group at the University of Edinburgh and also King's College London. And in this very elegant paper, authors, including Stephen Williams and John Whittaker, conclude that in patients after STAR with 25 Gray single fraction, we observe reduced local myocardial conduction velocity. However, this is in opposite to the previous reports from David Chang group, who showed that STAR actually shortened QRS duration and increased conduction velocity. Please note, however, this study was performed in mice and the whole heart was irradiated. In this study, performed in pigs, there was significantly longer local ERP and longer VT cycle length after STAR, but radiation was delivered to a very specific region of the left ventricle. In human, based on the NCOVT patient cohort, we can observe actually very mixed impact of STAR on QRS duration. In some patients, the QRS gets shortened, sometimes it's longer, in some patients, in one patient unchanged. Of course, we have to be very careful with interpretation because QRS duration is not a one-to-one equivalent of conduction velocity. But to conclude, it's still unclear how and why STAR works. So basically, this is still where we are. And using this graphical metaphor, you know it very well, the iconic Dark Side of the Moon album cover by Pink Floyd, we could say that the radiation beam, which at first appears monochromatic, actually contains many hidden layers and complexities. Just like the prism you can see here, the prism reveals the spectrum of light actually. We need to write tools and resolution to uncover and understand the full range of radiation effects on the heart. And I'm very, very happy. We continue studies on the conduction velocity using the OpenEP platform and also EP Workbench developed by Dr. Stephen Williams and fantastic team of people. And these are logos of partner institutions working on OpenEP and EP Workbench. And as a fellow at King's College and Royal Brompton, I could meet people behind these logos. And here I would like to express my special thanks to Stephen Williams, to John Whittaker, Vias Marquides, Vinush, and Arno, Ali, Neil, Magda, Castidius. So thank you very much for the opportunity to work with you on this. And also let me show you how we can use OpenEP and EP Workbench to analyze conduction velocity. Here you can see what we work on actually within the StopStorm project, collaborating with ICAM in Prague, University of Lausanne, Charité in Berlin, and Amsterdam University Medical Centers, and also with the involvement of Harvard University. We collected EP maps performed before and after STAR. How do we process the data? We start by using 3D slicer to segment irradiated ventricle, which you can see in greenish color here on the image. Then we reconstruct actually the 20 Gray isodose to visualize the area that received high dose radiation, represented here by the kind of a red shape. Then we can export these models to VTK format, which is readable, can be imported into EP Workbench. Next, using OpenEP, which works in MATLAB actually, we convert pre-STAR and post-STAR Carto exports to MAT format, and MAT format is also readable by the EP Workbench software. Next, we import ventricle geometry 20 gray isodose VTK models and also EPMAP into Workbench, where we merge these data, as you can see on the figure here. And based on this 20 gray isodose, we can draw, exactly 20 gray on the cartomap to get statistics on conduction velocity in the target area. Then we can save it as CSV file for further statistical analysis. And here you can see actually overlapping pre and post star maps. One is a little bit lighter and the second is a little bit darker. And that's how we can quantify conduction velocities in the target areas. Exported to CSV, we have numerical values, which then can be simply compared with standard statistical tests. And we hope that this will help us to understand how and why STAR works. And this analysis is in progress, and we hope to submit an abstract to ERA 2026, especially there will be dedicated stop storm session. And to confirm that EP Workbench is a real research tool, please see that we can adjust many settings. For example, conduction velocity method analysis, very relevant to what I described in terms of the project before. And this publication exemplifies that our group tests and implements the most precise and up-to-date algorithm into EP Workbench. Concluding, for now, you can find any color you like behind the STAR treatment, and STAR seems to be very, seems to activate distinct pathways that alternate conduction velocity, but further studies are definitely needed to understand more. And I truly, to truly understand the wide range of impact of radiation on the heart, we need the right tools and the right resolution. And I believe that OpenEP and EP Workbench are the one. Thank you very much.